Yeah, so we do want to address exactly the question that was raised. So one way to think about it, right, is that there's kind of a major conceptual elephant in all of systems neuroscience, which is the following. How is it that we can record order, say, 100 to 1,000 neurons in regions deep within the brain, far from the sensory motor periphery, and actually obtain scientifically interpretable results that relate neural activity to behavior and cognition? Right? This is actually quite remarkable considering that these brain regions contain about 10 to the 6th to 10 to the 9th neurons. So about 5 orders of magnitude more neurons than we currently record. So a question is, how has systems neuroscience been as successful as it has been in such an undersampled measurement regime? Or is it worse? Are we completely misleading ourselves? By recording so few neurons, are we getting a dramatically different view of the brain than we might get if we recorded all the neurons? Um, so that's what we'd like to address. Now, lest you think that there's just an experimental solution to this problem, while it is the case that recording technologies enable us to record more and more neurons, in fact, the number of neurons we can simultaneously record has been growing exponentially. So this is a type of Moore's law type of growth. But the doubling time is about 7 years, right? So right now we can record 100 to 1,000. So it would take about 120 years to get 5 orders of magnitude more neurons to fully record entire neural circuits in complex mammalian brains controlling complicated behaviors. So we're going to be in this undersampled measurement regime for the foreseeable future. And so what we need is we need a theory of neural data analysis that tells us how and when statistical analyses applied to a small subset of neurons reflect the collective dynamics of the much larger unobserved circuit in which they're embedded. So that's the question we'd like to address. Okay, so for the outline of the talk, we'll start with kind of trial average data. How do we, and I'll talk about what I mean by all this stuff. Trial average data, how do we accurately recover the dimensionality and shape of trial average neural dynamics? And we'll uncover this mathematical quantity, which we call neural task complexity, that determines both the dimensionality and recoverability of neural state-space dynamics. Then we'll move on to single trial data analysis. How well can we decode information from the brain on a moment-by-moment basis at each instant of time? And we'll find these phase transitions in our ability to decode information and accurately fit neural networks as a function of the number of recorded neurons and recorded time. And then at the end, I'll discuss an algorithm, which we call tensor components analysis. It's a method to perform multi-timescale neural dimensionality reduction. So neural dynamics exists on multiple timescales, right? On the dynamics of hundreds of milliseconds mediating cognition, behavior, action, perceptions, and much, much longer timescale, hours to days. That kind of dynamics mediates learning. So this algorithm can kind of simultaneously reduce dimensionality across neurons, individual trials, and time across trials to recover so-called cell assemblies with coherent rapid dynamics reflecting cognition and slower dynamics reflecting learning. So that's kind of the outline of the talk. So let's jump in. How many of you have experience with neuroscience before? I'm guessing not many of you, right? Okay, so small fraction, okay. So again, just interrupt me with questions like, whenever you want. And it's pretty informal, all right? So here's an example of a neuroscience experiment. This is the type of experiment from which we'll be analyzing data, right? So this is data from Krishna Shenoy's lab here at Stanford. And he studies motor control. So the types of experiments that he does is he watches monkeys move their arms, and he records from the motor cortex of the monkey, and he tries to relate neural activity to behavior. So here's an experiment where a monkey is holding a central target, then a reach target appears, okay? And then the monkey is supposed to plan a reach to that target, so you can look at motor planning in these types of experiments. But then the central target disappears. Okay, that's the signal that the monkey should start reaching. And then the monkey performs a reach, okay? What happens is neural activity during the planning period stays largely stationary over time. It doesn't change much over time. But the pattern of activity is indicative of where the monkey's going to move, right? But all the action is happening here within this about 600 millisecond window. It takes about 600 milliseconds for this monkey to do this rapid reach. And there's a lot of dynamical neural activity patterns during that 600 milliseconds across all the neurons. Okay, so they're recording from hundreds of neurons using an electrode array inserted into motor cortex and a slightly more upstream region, dorsal premotor cortex. Okay, they can record about 109 well-isolated single units or single neurons by measuring the extracellular voltage coming from these neurons, and that signifies when these neurons spike, right, or emit action potentials. So here's some examples of individual neuron firing rates. You can take the spikes and smooth them with a small Gaussian window and get smooth firing rates, and then you can average these over trials. And this is what you get for, say, one neuron, right? This is the neuron's activity pattern over time, over the 600 millisecond window, across different reaches in different directions and at different reach speeds and extents. Okay, that's just one neuron. Here's another neuron. It looks sort of completely different. Here's a third neuron. It looks extremely different as well. All right, and there's about 100 more neurons like this. So these data sets are somewhat complicated, right? There's a lot of complexity in this neural dynamics, seemingly. And so the question is, how are such data sets analyzed, right? So just a very simple approach that a lot of neuroscientists take is you measure the simultaneous dynamics of about n neurons during a task. And then you find, just say, for example, using principal components analysis, that the dynamics lives in quite a low-dimensional space, in the ambient 100-dimensional space of recorded neurons. So remember, in principal components analysis, you take every neural activity pattern across neurons that occurs in any instant of time, and you decompose it into a set of basis activity patterns. And you decompose it using a time-dependent set of weighting coefficients. That's just PCA. And so you can visualize the dynamics in this low-dimensional state space by the amplitude of each pattern present. And then you get this neural trajectory. And what a lot of works have found is that you can explain a lot of the variance in neural data using a relatively small number of dimensions, in this case about 10 dimensions, despite the fact that you recorded 100 neurons. Okay. So there's lots of work in neuroscience that recovers this kind of state space dynamics. And interestingly, the state space dynamics yields a remarkably insightful kind of dynamical portrait of what computation the circuit is doing. So for example, these are the state space dynamics of a locust antennal lobe when it's exposed to two different odors. And as you can see, the locust antennal lobe, the transient dynamics of the system contains a lot of information about the odor long before the dynamics reaches a respective fixed point, which is odor dependent. So it's processing information on the transients. This is data for a monkey prefrontal cortex, where the monkey's trained to detect different frequencies on its arm. And you see that most of the variance in the neural data accounts for the passage of time in the task, rather than the different frequencies, which is sort of an interesting finding. This is data for a monkey prefrontal cortex during a context-dependent decision-making task. And it turned out that in this work, done actually by Krishna Shenoy and Bill Newsom, done by their labs, the shape of these neural trajectories revealed an algorithm for how the monkey is making decisions. And also, you can record from about all 80,000 neurons in the zebrafish using calcium imaging, and you can fit that entire data set in about 3 dimensions, and you're okay. You don't lose much variance. So anyways, this is kind of a modus operandi for computational neuroscience with these large data sets. You recover the state space dynamics, and it's usually often quite insightful and quite simple. Okay? But this raises some fundamental conceptual questions. Again, can we trust these dynamical portraits of circuit computation despite recording so few neurons? How would the shape of these portraits change if we recorded more neurons? Did we even get their shape right? We make scientific inferences based on their shape. Did we get the shape right? Why is their dimensionality so low? What is the origin of the simplicity? Would it increase if we recorded more neurons? And more generally, what, if anything at all, can we learn about large dynamical networks at such an overwhelming level of undersampling? And so basically, can we obtain a predictive theory of experimental design that can tell us how many neurons we should record as a function of the complexity of the task or something like that? Okay. So I don't have the picture. So I should say this was work done by a grad student in my lab, Peiran Gao. Okay. So let me just give you the take-home message of the first third of the talk, which is basically we define this new mathematical quantity called neural task complexity. It takes into account 2 distinct sources of information, the sort of complexity of the task the monkey or animal is asked to do, and the smoothness of neural dynamics. And we prove a theorem saying that the dimensionality of neural data is upper bounded by this neural task complexity. Then we show that motor cortical data is actually as high dimensional as possible given this neural task complexity, which implies that in future experiments, if we record more neurons without an increase in the complexity of the task, we won't necessarily get richer data sets as measured at least by their dimensionality. And moreover, We can recover conditions for accurate recovery of dynamic portraits. We can show that the act of neural measurement is like doing a random projection. I was curious, how many of you are familiar with random projections? This is the CS department, so. Oh, again, a surprisingly small fraction. Okay. Okay. We'll go over random projection theory, but it's a very beautiful theory. We can show that what neurophysiologists have been doing for the last 50 years is essentially doing a random projection, which is kind of an interesting connection. And then based on this, we can show that past results, i.e. existing dynamic portraits, are likely to be accurate despite recording so few neurons. Okay, okay, so what is this? Oh, I don't have the. Okay, it's okay. Well, so what is this neural task complexity? So we can think about the manifold of neural states that are explored by the experiment as an embedding of the behavioral manifold into neural activity space, right? And the basic idea is here's a monkey reaching to 8 directions, okay? And you have some reach extent or reach time, okay? And you trial average all of this. So the manifold of behavioral states explored by the monkey is roughly a cylinder parameterized by the angle of the reach and the time into the reach, okay? Or duration of the reach. Okay, now this trial average, this manifold under the trial average neural dynamics gets embedded into some kind of complicated cylinder that we don't really understand fully, okay? But we can characterize the complexity of this cylinder by 2 things. Roughly the volume of the behavioral manifold, which is the numerator of this quantity. So let's say you have a general manifold that has k intrinsic coordinates, right? Here it's 2 intrinsic coordinates in this example. One is theta, one is 2 pi, or the solid angle, and one is the duration, the length of the reach. So this has a certain volume, but that gets embedded into the space, and there's some volume of a unit cell of how much you move before things curve. It's sort of like an autocorrelation length. So let's say that I moved in this direction, or let's say that this section or cell of the manifold roughly got matched to this section or cell of the manifold where there wasn't much change in the curvature. Then this entire section of the manifold roughly explores only 1 dimension in firing rate space. But then let's say you have to move a certain distance along each dimension of this manifold to get to another cell that explores a different dimension, right? So then roughly the total number of dimensions that could possibly be explored is the volume of the behavioral manifold divided by the volume of the unit autocorrelation cell over which the manifold doesn't really curve that much, okay? So just intuitively, this is what's the measure of neural task complexity. Our theory gives us a way to extract these autocorrelation lengths, and of course the experimentalist determines the volume of the behavioral manifold that's explored. And so this ratio is the neural task complexity, and there's a constant involved that's also given by the theory, right? So roughly, you can prove a theorem that says that the neural dimensionality is upper bounded by the minimum of this neural task complexity measure in the number of recorded neurons. So at least at a high level, this should be overall intuitive. A manifold that has larger volume, i.e. bigger numerator, and is more wiggly, i.e. smaller denominator, can potentially explore more dimensions, okay? All right. So now let's go back to the neural data. In primate motor cortex, again, there are about 100 million neurons controlling 650 skeletal muscles. And in these experiments, we recorded about 100 neurons. The PCA dimensionality, which is roughly the number of principal components you need to explain 70% of the variance, across all 8 reaches is about 7. The dimensionality to explain just one reach, the variance in neural activity across one reach, is about 3.3. There's a particular measure of dimensionality that we're using it's something called the participation ratio of the eigenvalue spectrum of the neuron by neuron covariance matrix, which roughly measures how many principal components are active, okay? And a certain participation ratio for a wide variety of spectra corresponds to how many PCs you need to keep 70% of variance explained. It turns out that 70% is not an important number. We could multiply the participation ratio by a constant fraction, and we could get up to 90%. It would be the same exact theory. Okay? So, so then again, the puzzle is these numbers are much smaller than the total number of neurons. And again, why is that? What's limiting the complexity? Have we found something intrinsic about brain dynamics that makes it simple? Or is the simplicity of brain dynamics a consequence of the simplicity of the task the monkey is asked to perform? Okay, so it turns out the latter is true. If you compute the task complexity, the neural task complexity, it's about 4.2 for a single reach and 10 for multiple reaches. Okay, so as you can see, the neural dimensionality is close to this upper bound. Okay, so this suggests, so the implication is the neural dimensionality is not necessarily small, but it's almost as large as possible given the simplicity of the task. And this makes certain predictions. If you vary the complexity of the task by varying the duration of the reach, or equivalently the amount of time of the reach that you analyze, then if indeed the dimensionality of the data is limited by the complexity of the task, then the dimensionality should vary linearly with that time that you keep. Okay, that's prediction number one. Prediction number two, the number of neurons in the data set is kind of irrelevant in this case. If you vary the number of neurons in the data set, the dimensionality should roughly be unchanged, right? Because it's the complexity of the task that's limiting the dimensionality. So it doesn't really matter that you measured 100 neurons. If you reduce the neuron count, you shouldn't change the dimensionality. Okay? So prediction number one is verified, right? As you vary the task complexity by varying the duration of the reach that you analyze, you find that the dimensionality increases with the reach duration. It hugs this kind of dimensionality upper bound set by the complexity of the task. And two, as you drop even 60% of the neurons, the dimensionality remains largely unchanged. Okay? So, okay. Whoops. Okay, so that's the most boring part of the talk. The next most interesting part is what about the shape of these trajectories? Dimensionality is one thing, but did we get the entire shape of these trajectories correct? Okay? So what, just to introduce the idea, is what is an intuitive situation where you could record a subset of neurons in a circuit, but still get its state space dynamics correct? Right? So here's an example. Here's the simplest visualizable example where you have 3 neurons. So this is the firing rate of neuron 1, firing rate of neuron 2, firing rate of neuron 3. For whatever reason, let's say neural activity patterns that are explored during this experiment live on a one-dimensional manifold, and it's curved but not too curved, okay? And it's roughly randomly oriented with respect to the single neuron axes, okay? So then it's kind of clear that even if you don't measure all 3 neurons, let's say you only get to measure 2 of the neurons, you see a shadow of the full state space dynamics of the brain onto the coordinate subspace spanned by the 2 neurons that you do get to measure. And you can see that it doesn't matter which 2 neurons you measure, you get the same answer, and that answer is roughly similar to what you'd get if you recorded all the neurons. Okay? Of course, life doesn't need to be that rosy. It could have been the case that, for example, neuron 2 were doing all the work, so that this manifold is axis aligned to the single neuron axes. And then, of course, if you only get to record a subset of neurons, that subset better include neuron 2, right? So this is kind of a pessimistic picture, okay? This picture will arise if neural activity patterns are distributed across neurons, okay? Okay, so let's actually hope for the best and hope that the brain is in this regime and not in some other regime, not in this regime, okay? Then let's pursue the theoretical consequences of the brain being in that regime and derive some theoretical consequences, then test those consequences in the actual brain to see if this regime is a good enough approximation, okay? So this is a very simple picture to analyze, but how do we make it quantitative and think about it in much higher dimensions, where the number of recorded neurons is 100 to 1,000, the dimensionality of this manifold may be higher than 1, and so on and so forth, right? So how do we generalize that? Well, let's say that this neural manifold is indeed randomly oriented with respect to the single neuron axes. That's a statement that can be made precise, but we'll just keep it intuitive for now. Then, an experiment that we can do, i.e. measure a random subset of M neurons, becomes equivalent to an experiment we cannot yet do, which is measure M random linear combinations of all neurons. So imagine you have a magical electrode that measures a random weighted sum of all the neurons, another magical electrode that measures another random weighted sum of all the neurons, and you have m of these electrodes, right? Okay, so why are these two experiments completely equivalent to each other if the neural manifold is randomly oriented? Well, this experiment corresponds to projecting the neural state space dynamics onto a coordinate subspace, whereas this experiment corresponds to projecting this neural dynamics onto a randomly oriented subspace, i.e. it's a random projection, okay? So if the neural manifold is randomly oriented with respect to the coordinate subspaces to begin with, then projecting onto a coordinate subspace and projecting onto a random subspace are equivalent to each other, right? So if this assumption is true, then the act of neural measurement is equivalent to the act of random projection, okay? So let's assume that's true for now, okay? And let's derive consequences. So now let's review the theory of random projections. It's really a beautiful theory that underlies compressed sensing and a lot of ideas in signal recovery in an undersampled measurement regime in computer science and EE and signal processing. So what happens often is, of course, signals that we're interested in live in a high ambient dimensional space, say images. The ambient dimensionality is the number of pixels for grayscale images. But of course, the signals of interest don't explore this full high dimensional space. They live in a much lower dimensional space. For example, sparse signals live in a union of coordinate subspaces. You could have a finite number of points, or you could have a smooth manifold, okay? Now, let's say that you randomly project this space down to a lower dimensional space using a random projection matrix. So you're projecting n-dimensional space down to m-dimensional space. So A is an n by m matrix. S is a signal of interest. In our context, it's the set of neural firing rates of all the neurons in the brain region. And X is the set of neural firing rates of m recorded neurons, okay? Again, the neural activity patterns that we observe live in some low k-dimensional submanifold in n-dimensional space. And a critical question is, when will the geometry of this manifold be preserved under a random projection? By the preservation of geometry, we mean the following. Let's consider 2 points in the high dimensional space and consider the distance between them measured under a Euclidean metric in the high dimensional space. So that's SA minus SB, for example. Then you project it down to the low dimensional space, and up to a normalization, you measure the distance between the 2 points in the low dimensional space. So this is the fractional error in distances measured in the high dimensional space relative to the low dimensional space, okay? Now this is for 2 points. Of course, you don't want to just preserve the distances between 2 points. You want to preserve distances between all possible pairs of points, but not in the full space, only on the manifold that you care about, okay? So we can define the distortion as the max over all pairs of points of this DAB measure. Let's call that epsilon. So epsilon is the worst case distortion over all pairs of points, okay? Epsilon's a random variable because your projection is a random matrix, okay? Or a random subspace, okay? So there are theorems about the behavior of epsilon and its scaling with the number of dimensions and with n, k, and m, basically, okay? This is the most important theorem that we'll invoke. This is work by Richard Baranyuk and Michael Wakin. Actually, these are old slides. We have some work that sharpens these results and gets better bounds. It's on our website called Random Projections of Random Manifolds. But basically, the theorem is the following. As long, and I'm going to use neuroscience language here, okay? As long as the number of recorded neurons m, or the dimensionality of the subspace, is bigger than, up to an order of magnitude, 1 over epsilon squared times k, the intrinsic dimensionality of this manifold, times the logarithm of the curvature times the volume of the manifold, okay? Then the worst case distortion is indeed order epsilon with very, very high probability over the random choice of projection, okay? So this is interesting. The ambient dimensionality, in our context, the total number of neurons in the brain region, doesn't matter at all. What matters is the intrinsic dimensionality of your manifold and its curvature and volume, okay? And of course, the distortion level that you'd like to get, all right? Okay, now this actually makes a striking prediction, okay, that we can now test in neural data. Part of the business of a theoretical neuroscientist is taking theory and coming up with a way to test it in neural data. So this is the prediction, okay? If you convert this random projection theory into neuroscience language, it's saying the number of neurons you need to get a certain distortion is proportional to the logarithm of this neural task complexity. Remember, neural task complexity was the volume of the manifold in the numerator times the curvature, or 1 over the volume of an autocorrelation cell. So actually, this log of C times volume mathematically becomes the logarithm of the same neural task complexity measure that upper bounded dimensionality. Okay, so what this is saying is it's saying that if you were to reduce the number of neurons or change the complexity of the task, it's saying that the isocontours of constant distortion form a straight line in a plane spanned by the number of neurons you record and the logarithm of the complexity of the task. In our case, where our task is just reaches along a single dimension or reaches along a single axis, the complexity of the task is the duration of the reach. Okay? So it's a logarithm of time. Now, of course, we'd like to record all of the neurons in the motor cortex under the most complicated behavior possible, but we don't have that. We have a data set with a finitely complex task, and we have about 109 neurons. So what we can do is we can, again, vary the complexity of the task by varying the amount of data that we analyze. And we can subsample neurons. And we can recover the state space dynamics of the circuit using a smaller number of neurons for any given task complexity and compare its distortion relative to that which you would get if you took all the neurons that you recorded. Okay? So you will get some distortion. It'll vary as a function. But what this is saying is on this weird plane that's linear in the number of neurons that you keep, but logarithmic in time, if you vary along this axis, along a straight line, you'll get the same distortion. And let's say you allow yourself to be more generous. For a given task complexity, you allow more neurons. You'll get a lower distortion. But again, there'll be a different isocontour of distortion that's, again, a straight line. Okay? So this makes now a testable prediction. We can actually do this in neural data from the monkey's brain and actually plot these isocontours of constant distortion. And what you find is that the prediction is strikingly verified. So this is, again, the number of recorded neurons that we keep. This is the neural task complexity. This is a logarithmic axis. This is a linear axis. This is the distortion level, and the isocontours are indeed straight lines. Now remember, this isn't just an empty mathematical test of a theory for which we already have a theorem, i.e. random projection theory. This is a test of the very assumption that random projection theory constitutes a good model for the process of neural measurement. Okay? And it turns out it does seem to constitute a good model for that process, at least as measured by this prediction, or as assessed by this prediction. Okay? All right, so that's the first third of the talk. Again, let me just summarize. So basically, whenever you look at the dimensionality of neural dynamics in the brain, to ask if it's complicated or not, what you really want to do is compare the dimensionality to the neural task complexity. If it's as high as that, then your dimensionality is constrained by the complexity of the task and the smoothness of neural data. Only if it's much less than that have you discovered additional intrinsic dynamic constraints in the data beyond task complexity and smoothness. And also, this random projection theory actually yields a surprisingly optimistic message for neuroscience, is that in many, many cases, because the task isn't that complicated, the neural manifold doesn't have that high volume, is not that curved, we can get away with recording a very small number of neurons. Okay? So that's really good news, actually, for neuroscience. And as we start doing more and more complicated tasks, it turns out that the number of neurons we simultaneously record is not really the bottleneck. It's exploring the manifold that becomes the bottleneck. And this actually impacts the design of technology in neuroscience. You would much rather have technologies that allow you to stably record neurons for long periods of time as you go to a more complex task, because the time it takes to explore a manifold grows exponentially with the dimensionality, intrinsic dimensionality of the manifold. But the number of neurons you need to record grows linearly. Okay? So considerations like this can drive the design of technology as well in neuroscience. Okay, so let's move on to the second part of the talk. Yeah, sure.
So how do you measure the distortion in practice? Because you don't know the distances in that full n-dimensional space.
Oh yeah, so we do it in the 109-dimensional space, compared to like m-dimensional space where m is less than 109.
And the 109 is where the number of neurons you have.
Exactly, yeah.
And
So it's as if the entire brain is 109 neurons.
Yes, I know. So how does that relate them back to the actual?
So remember, the ambient dimensionality really doesn't matter, right? So even if you had like 10 to the 6 neurons, you'd get a similar answer because the volume of the manifold is so small and its intrinsic dimensionality is 2, right? So if you have like
But that's an assumption, right?
No, that's a consequence of the theory. So so what's theory and what's assumption, right?
Yeah.
So if the random projection theory is a good model for the active electrophysiology measurement, given that assumption, we expect the exact same answer if we did the analysis I did with 109 neurons versus 10 to the 6 neurons, given the intrinsic dimensionality of the manifold is only 2, right? 2 is much, much bigger than 109.
That's what I'm asking too.
That's
an assumption that the manifold is two-dimensional.
No, that's not an assumption. That's the trial average manifold.
We don't know what it is, right?
Huh? Oh, okay, so for this...
the way you design your experiment then.
Yeah. Only for this experiment, right? So for other experiments, there will be a different dimensionality. But then you plug in that dimensionality into the theory and you get an answer for how many neurons you need.
If you take a subset of your 109 recorded channels, or multiple randomly chosen subsets, how much variability is there if you perform this analysis on the subsets?
Between subsets? Yeah, so the distortion between subsets, it's similar to the distortion between any subset and the full set, right? Because of triangle inequalities in geometry. It's a similar answer. Actually, I didn't even include this because this is even more recent work. This actually has a really cool practical implication. So when people record like hundreds of neurons, they stick an electrode array into the brain. Now each electrode could be listening to multiple neurons. So there's a complicated clustering problem. Each neuron leaves a slightly different waveform on the electrode. And so in order to get individual neurons, you need to solve a clustering problem. It's called spike sorting, right? Spike sorting is the bane of existence of experimental neuroscientists, right? Because you're measuring random linear combinations of neurons in your electrodes, and you need to demix that random linear combination. It turns out we have a separate paper under review and also in the bioarchive called Accurate Recovery of State Space Dynamics Without Spike Sorting. This theory says that what I've told you, one consequence of this theory is that if you record 100 neurons out of 109, you get the right state space dynamics. This also says if you don't do spike sorting, but you just look at the high frequency activity on the electrodes and just do dimensionality reduction directly on that without spike sorting, you get the same state space dynamics. So what this does is it unlocks like a whole bunch of data sitting on hard drives that people have not spike sorted, and it makes that available for analysis, at least at the level of state space dynamics. So that's another paper that's on our website and is under review as on bioarchive. So that was done by Eric Troutman as the lead author in Krishna Shenoy's lab in a collaboration between my lab and his. So, and by the way, this is also part of the reason why brain-machine interfaces work, why you can decode at least simple movements from the brain by only recording 100 neurons. Of course, if you try to decode more complicated movements, you probably have to record more neurons. But this is going into single trial data analysis, which because you need to, in a BMI, you don't want to recover the trial average trajectory of the arm. That doesn't really mean anything for a patient. You want to decode the instantaneous intentions of the patient and translate them into the action of the prosthetic device. So that's why single trial data analysis is, of course, extremely important. So let's move on to that, unless there's any other questions on the first, third. Okay. So what do I mean by a single trial theory, right? So here's kind of the setup, okay? We always have limited experimental resources. The resources are M is the number of neurons we can simultaneously record. P or T is the number of training stimuli or amount of time we can observe the brain. Okay, let's say you're observing, let's say in a brain machine, to design a brain machine interface, you're watching a monkey, for example, reaching multiple times. On each reach, you record the individual trajectory of the reach and the individual neural trajectory in the brain. right? And you do this many times, and you build up a dictionary, so you get a certain amount of training data. Then subsequently, on held-out data, say a single trajectory from the monkey's brain, neural trajectory from the monkey's brain, you'd like to construct a brain-machine interface that decodes the trajectory the monkey wants to move its arm on a single trial, right? So P or T is the amount of training data you have for subsequent single trial analyses. And of course, there's always some signal-to-noise ratio or trial-to-trial reproducibility in our data. Even if you show the same visual stimulus to a brain, neural activity patterns will vary from trial to trial. It's a major open question whether this variation is irrelevant noise or it's just the rest of the brain thinking. And therefore, it's very interesting cognitive variables that are changing from trial to trial. Okay, then you also have some measure of the complexity of the experiment or the neural data. So some measure of the complexity of the stimuli or behavior or latent variables, latent cognitive variables or thoughts or manifold of visited neural states. So this is some complexity measure. So then the question is, given these limited experimental resources and the complexity of the thing we're trying to decode, or the neural data, how well can we decode behavior on single trials, and how well can we learn the structure of unobserved latent cognitive variables contributing to this trial-to-trial variability in the brain? Okay. We'd ideally like to have a theory to help us design experiments before they're done. Okay. So let's consider, for example, a very simple, relatively simple warm-up problem, which is inferring latent subspaces in which data live. So here's the idea. Let's say that on individual trials, there's some latent space, like a low-dimensional space of things that the monkey can think, movements that the monkey can imagine, or even stimuli that the monkey sees, in which case they're not latent variables, they're actually observed variables. Okay, so there's some low-dimensional space, and on individual trials given by these white dots, these latent variables take different values in this low-dimensional space. Okay? Now, in the monkey's brain, this is the full n-dimensional space of all the relevant neurons in the circuit. For each of these latent states, there's a different neural activity pattern. And we can imagine just a simple linear embedding, for example. Again, if it's a nonlinear curved embedding and the curvature is not that high, then these low-dimensional, not too curved manifolds live within a subspace of dimensionality not much higher than the intrinsic dimensionality of the curved manifold. Okay, but then again, there's noise. There's trial-to-trial variability in the data. So you're not always living directly on the manifold. You're living off the manifold. Okay? Now, so that's the state of the brain. That's the assumed state of the brain in this very simple model. Okay? Now, what do we get to observe? Again, we don't get to observe all the activity in the brain. We get to observe a subset of neural activity patterns. Okay, and we only get to observe the noisy activity patterns. We do not get to see the latent subspace. And we do not get to see the clean single trial latent variables on trial to trial, right? So given only these x's, can we infer both the orientation of this latent subspace and on individual trials, can we denoise the data and extract from the M recorded neurons the latent variable that was driving neural activity, okay? Whether it's internal cognitive states or motor actions or stimulus-evoked responses, okay? So again, the quantitative parameters, you have n neurons, n total neurons in the circuit. You only get to record m of them, and you only get p stimuli or p latent states, okay? And let's say the stimuli are encoded by this k-dimensional linear subspace, okay? Then what regimes of m, n, p, and k in the SNR can we correctly recover both the subspace and its dimensionality? Right? Okay, so we can formalize this algebraically as a low-rank matrix denoising problem as follows. So let's imagine that the k stimuli or k latent variables that you see can be arranged, the p latent variables that you see, each of them is a point in a k-dimensional space, so they can be arranged as the columns of a k by p matrix. So each column is one latent variable, and these are the different trials that you see, okay, p trials. Okay, these latent states are embedded into the brain through some embedding matrix or in some subspace, and I'll describe that subspace by the columns of an orthonormal matrix, okay? This is an n by k matrix. Now again, you don't get to observe all n neurons. You only get to observe a subset of them, and this is the random sampling operator. which is each row is just one neuron is active, has a 1, and the rest are 0, right? So each row tells you which neuron you're observing, and there's m neurons, okay? Plus, you have observation noise, which I'm just going to assume is IID across neurons and trials. It's kind of an idealization of the problem, all right? And so this is your data. Your data is the matrix S times U times X plus Z. And what you'd like to do is from this data, recover U, or at least the projection of U onto the recorded subspace, and for each individual trial, recover the columns of X. So X is hiding inside your data, and you'd like to recover it, okay? What do I mean by the SNR? By the SNR, I mean the signal-to-noise ratio of individual neurons. So if I were to trial average, right, for different latent variables, I'd get a different firing rate. And if I take the squared, the power in the trial average firing rate, that's the variance of the signal. But then if I condition on the latent variable and look at the variability condition on that, that's a single trial noise. And that has a variance sigma squared n. And so the SNR is, as usual, in EE, the variance of the signal to the variance of the noise. Okay, so I have a well-defined problem. In the interests of, how many of you really like math and theory here? Okay, interesting. There are many who are willing to admit that they don't. All right. So, yeah, good for them, yeah. So I love math and theory, but I'm going to break my heart and hopefully not piss off a lot of people. And I'm going to skip the theory. But it involves really interesting random matrix theory and low-rank matrix denoising and high-dimensional statistics and so on and so forth. But I'm going to tell you the answer. The answer is the following. As long as the product of the signal-to-noise ratio and the square root of the number of recorded neurons and the number of training stimuli that you have is bigger than the dimensionality of the latent subspace, right? And of course, two extra constraints. The number of neurons must exceed the dimensionality of the latent subspace, and the number of trials must exceed the dimensionality of the latent subspace. But as long as these three conditions hold, then you can do what I said we would like to do. We can accurately recover these unseen variables. We can accurately recover the orientation of the subspace, and then subsequently on single trials, if I give you a new X here that you've never seen before, because you know the subspace, you can project it down onto the subspace and recover the latent variables. Okay? So this, again, makes a striking prediction about neural data. Okay? It says that there's an interesting hyperbolic trade-off between two very different experimental resources: the number of neurons that you record and the number of trials that you take. Because your ability to do single trial data analysis only depends on the product. So you can always trade them off against each other. Okay? So, so what this says is, well, we can do this in neural data. Again, neural data collected from Krishna Shenoy's lab. So it again says, so now K has been replaced with D in this slide, but it's the same concept. It says there's a hyperbolic phase transition boundary in the M by P plane, number of recorded neurons and number of trials. Okay. So we confirm this in simulations. So here we simulate random smooth neural trajectories with an SNR, a very small SNR of 0.02, living in a 4-dimensional space, and we ask how many, we subsample a number of neurons, we subsample a number of trials, and we ask how well can we recover the dimensionality of the subspace and the orientation of the subspace. So you can define an overlap between subspaces where it's 1 if they're identical and 0 if they share no common dimensions. They're completely orthogonal. It generalizes the cosine of the angle between two one-dimensional subspaces. Okay? And so as you can see, there is this hyperbolic phase transition boundary in simulated data, but that's just a confirmation of our theory. Okay? The question is this theory at all relevant to the brain? Because the brain isn't making the IID assumptions that we're making. It isn't making a linear subspace assumption that we're making. But we expect that qualitatively the structure should show up, partially because of what we know about universality and random matrix theory. we used random matrix theory to derive this hyperbolic phase transition boundary. And a lot of results in random matrix theory doesn't depend in detail on the distribution of individual elements in that random matrix. So emergent properties like the eigenvalue spectrum and so on and so forth don't depend on detailed distributional assumptions about individual matrix elements. They do depend on having weak correlations between the matrix elements. But in any case, we can test this in a monkey. So what we did was we again looked at data from the Shenoy lab, the same data that I talked about before in a trial average setting, but we look at them in a single trial setting, right? And we use something called Gaussian process factor analysis. It's basically a souped up version of PCA with some smoothing along the temporal direction to extract latent trajectories, neural trajectories, in 147 trials for reaches to a single target, okay? And we use a different algorithm even from just low-rank matrix denoising. We infer the dimensionality explored by these latent trajectories using cross-validated Gaussian process factor analysis. So there's a parameter which is how many dimensions that you keep, and you can choose that parameter using cross-validation, how well you predict single trials on held-out data, and so on. The noise in the data is pink and temporally correlated. It's not IID in time and trials. So there's a different noise in the data, okay? But still, sorry for the resolution, you still see this hyperbolic trade-off between how well you infer the subspace, the dimensionality of the subspace, and how well you get the right subspace. Now again, this is a situation where we only had 109 neurons, so we infer the dimensionality of the subspace and the using all of the neurons, and we define the correct subspace to be whatever subspace we got using all of the neurons. Then and all of the trials. Then we subsample neurons and we subsample trials and we measure the dimensionality again, and we measure the subspace overlap between the subspace that we got using fewer neurons and fewer trials with the projection of the quote-unquote true subspace using all neurons and all trials onto the subspace of the subset of neurons that we keep, okay? But to make a long story short, you again see an interesting hyperbolic trade-off between neurons and trials. So this is kind of random matrix theory operating in the brain, right? There is this trade-off between two very different resources, neurons and trials. So in future experiments, it's really important to be cognizant of this trade-off, especially in the design of experiments before the data is actually collected. Okay, there's another super interesting question that we can ask in terms of this, what are we doing when we're doing neuroscience? So you're familiar with sort of deep learning and recurrent neural networks and things like that. And so what people are doing is they're, again, taking neural data, and they're recording a subset of neurons, looking at that dynamics, and fitting a recurrent neural network to that dynamics, okay? Now, what the heck is it that we're doing, right? How is it that a recurrent neural network fit to 100 neurons that all reflects anything about the dynamics of the circuit, using all the neurons? Is there anything about the circuit dynamics that we can get right? I.e., what the hell are we learning about the brain by fitting neural networks to the brain, used to a small subset of neurons? Okay, that's a super interesting question, right? Especially because everybody's doing it now, and it's not clear what it means when you do it, all right? So we also asked this question, okay? So consider, and we formalized it using theory, and we applied the theory to linear neural networks, but then we did simulations of nonlinear neural networks to show that the qualitative structure of the theory holds also in nonlinear neural networks. So what's the setup of the problem? We can formalize this problem as follows. So consider some high-dimensional neural circuit with n neurons. Again, you only need to record m of them. The notation's all the same for a finite amount of time t. What can we infer about the circuit dynamics when m is much smaller than n and t is not too large? And of course, in general, the answer is absolutely nothing. Okay, it's a seriously pessimistic problem in this regime for arbitrary recurrent neural networks. However, we might assume some underlying simplicity about the recurrent network dynamics. For example, for some reason, that the network dynamics may be forced to live in a k-dimensional subspace, okay, because of the recurrent connectivity. Now, where you imagine the recurrent connectivity is constraining the activity patterns to live in a low-dimensional subspace. Okay? So then the question is, for what regimes of M, N, T, and K can we correctly recover the dynamical properties of the circuit? So again, we'll work out the theory in a linear model. So this is a linear neural network with a decay time constant of tau. It's kind of a discrete time version. There's a recurrent connectivity, and this is a rank K matrix. Okay? So basically, without this matrix, activity would decay to the origin. But because of the rank K nature of this matrix, it's amplifying activity patterns, but only in K dimensions. Okay? And there's some noisy input that we're just going to take to be white noise here. Okay? Now again, you don't get to observe the full state. You only observe a subset. And this is your random sampling matrix. Okay? So now what do people do? They fit an autoregressive model to y of t. Okay? But what the heck does the autoregressive model have fit to y of t have to do with an autoregressive model fit to x of t when m is much, much less than n, when y is a much lower dimensional vector than x? Okay? So there's interesting methods for fitting these using subspace identification that leads to Hankel matrices. You do low-rank decompositions of these Hankel matrices, and there's some nice random matrix theory you can apply to these. But let me just tell you the answer, okay? It turns out that there's, again, a phase transition in the number of recorded neurons, the amount of time that you record in that plane, okay? That whenever this so-called Hankel matrix has a gap in its eigenvalue spectrum, then, so this is an example where you have 1,000 neurons, a rank 4 connectivity matrix, and this is kind of the time scale of single neurons, right? So now you get to measure for a time duration of 2,000. So roughly the number of observations is 2,000 divided by 9. That's the dimensionless ratio of the recording time to the time constant of the circuit. And then the number of neurons is 200, not 1,000. So you're recording 1/5 of the circuit. And then you ask, I fit an autoregressive model to Y, so I get an autoregressive matrix C, right? Oh, sorry. Sorry, I fit a latent variable model to Y, and I get this latent dynamics A that I get from a subset of neurons. You could just ask, did I get the eigenvalues of the full matrix correct, right? Does the 200 by 200 matrix that I get on my observed data have the same eigenvalues as the 1,000 by 1,000 matrix, okay? And the answer is, for this regime, yes, right? So the red are the true eigenvalues, and the x are the estimated eigenvalues, and you actually got the right answer. So despite subsampling, you recovered some aspect of the dynamics of the full circuit, i.e. in a linear system, the eigenvalues of the connectivity matrix. Okay, they're complex because it's not a symmetric matrix. On the other hand, if the Hankel matrix doesn't have a gap, there are errors in the estimate of the eigenvalues. Okay? So for example, with 50 neurons, you cannot do it, whereas with 200 neurons, you can, for the same amount of recording time. Okay? Again, so again, here's the phase transition, number of recorded neurons and duration of time. And there's a hyperbolic phase transition boundary that we get from theory. This is simulated data. The transition in the simulated data matches our theory. On this side, where you have the product of number of neurons and time exceeds the threshold, you get the correct eigenvalues. And here you get incorrect eigenvalues. Okay, that's kind of the take-home message. So it's a very similar story, a trade-off between two very different parameters, number of neurons and the amount of time you get to watch the circuit. Okay. Okay, we tried this on nonlinear neural networks, and we observed similar hyperbolic trade-offs that shift depending on the nonlinearity. Okay. So then the last part of the talk, which I think I can finish in about 10 minutes. Yeah. Huh? No, not the single trial's not out yet. Yeah, the single trial stuff's not out yet. Yeah. Okay. This stuff actually is out. The tensor components analysis. This is in press at Neuron, and it's on the bioRxiv and on our website. So this last part is actually a practical algorithm to solve the following problem. Okay. And this is work done by Alex Williams in my lab, who kind of spearheaded this work. So the goal is, how do we simultaneously reduce the dimensionality of data across, again, two very different dimensions? Time within trials, right? So remember, neural data is often organized into trials. Like the monkey does something, and maybe initially it doesn't do it well. Then it keeps doing it again, and over time it gets better and better at it, okay? And then, so now you have two very different timescale dynamics. Within a single trial, which could be a second, the monkey is doing something like reaching or something like that. The brain is having this churning activity that's correlated with the reaching. But then across many trials, this dynamics is slowly changing to reflect learning or performance or improvement, right? So how do we simultaneously obtain low-dimensional descriptions of both time with neural dynamics within trials, reflecting perception, action, decision-making, motor actions, cognition, and so forth, and time across trials, reflecting learning, and also there's other really interesting state changes in your brain, like how much attention you're paying can wax and wane from time to time. Your arousal state, your changes in cognitive state. Like monkeys could be thinking different things on different trials that are not under experimental control. And we like to extract all of that in an unsupervised manner. And especially changes in cognitive state that are not under experimental control must be extracted in an unsupervised manner because you don't have a covariate with which to regress against neural data to discover dimensions in neural activity space that encode that covariate, okay? So the idea we had was we treated these two disparate timescales as two distinct dimensions of a third-order tensor, with the third axis being neurons, okay? So here's the basic idea. So we kind of discussed trial average PCA at the beginning of the talk. So here you have neurons by time within trials, and you have the spikes of neurons. Then you have another trial and another trial. And let's say you assume that all trials are identical, then you just average all the trials and maybe smooth over time. So you get a matrix of neurons by time, which reflects the trial average firing rates of all the neurons. And you decompose it using PCA. So you basically do a matrix factorization where you write this matrix as an outer product of patterns across neurons and patterns across time, right? Okay, but that's assuming that individual trials are statistically homogeneous, right? It's assuming stationarity. Fundamentally, when an animal is learning, you cannot assume stationarity. In fact, learning is tautologically non-stationary, right? So trial averaging will make processes like learning and changes in cognitive state invisible, right? So one way to deal with that is to take these matrices and just concatenate them across time and then do PCA, right? So then you decompose it into a pattern across neurons, and then very, very long patterns across both time within trial and time across trials, okay? People sometimes do this, but it isn't very successful because there's so many parameters involved in this decomposition. Because there's nothing constraining, it's assuming that there's nothing common across trials, right? Each individual trial could be completely different. It's not assuming anything common. Whenever you're doing single trial data analysis, you're always making an assumption about something that's common across trials and something that varies across trials. Okay, and trial concatenated PCA is not making any assumptions about what's common across trials. So there's too many parameters, so it's often not that useful in single trial data. Okay? The approach that we used was to use something called canonical polyadic tensor decomposition. For neuroscientists, we called it tensor components analysis to rhyme with PCA. It's called like TCA. Anyways, but the technique, the mathematical technique that we're using is something called canonical polyadic tensor decomposition. It's a generalization of PCA in the following sense. You can take these neurons by time matrices and stack them into a third-order tensor, and then decompose this tensor into a sum of rank 1, outer products of rank 1 tensor, or sum of rank 1 tensors, okay? So each component gives you 3 factors. It gives you a pattern of activity across neurons, which you can roughly think of as cell assembly, or a pattern that varies coherently somehow, right? It gives you a temporal factor, which is a kind of dynamical mode shared by these neurons across time within trials, okay? And then it gives you a trial factor, which is just a scalar amplitude variable that scales up or down this pattern across neurons, and this pattern across time. Okay? So it's like a gain modulation factor, right? And this is just, it's just what it does. It's a completely unsupervised algorithm. You throw at the data tensor, it spits this stuff out. Okay? It turns out that this very simple off-the-shelf algorithm is actually surprisingly useful in neuroscience. So one way to think about this is completely equivalent to a very simple gain-modulated neural network, where let's say these are the neurons that you're observing or recording. You assume that there's some number of latent factors here corresponding to the number of outer products you have. Okay? You assume that each latent factor has its own temporal waveform. The shape of this temporal waveform is common across all trials. So that's the assumption of commonality across all trials. So this is the shape of one factor waveform, this is the shape of another, this is the shape of another. But what's changing across trials is the amplitude of each waveform. Okay? That varies across trials. So basically what you're doing is you're summarizing each trial. Let's say there's k factors. You're summarizing it by k numbers, the amplitude of each waveform. Okay? And that's what's driving the activity. And it turns out that this is, if you do CP decomposition on just observations of activity patterns and noisy versions of them, you can accurately recover all of these factors. Now, what's really interesting is PCA is fundamentally a subspace recovery algorithm, right? PCA only gives you unique principal components if you have an orthogonality assumption on your principal components. Without such an orthogonality assumption, you just recover a subspace if you keep k principal components. Here, the individual factors actually matter, right? This actually recovered the individual factors identically, and it doesn't require them to be orthogonal. PCA applied to different unfoldings of the tensor doesn't work. Independent components analysis doesn't work, but the CP decomposition works. Okay, that's kind of to be expected because we did it on data drawn from the model, and so it's to be expected. Yeah.
So, but the question is, that's the pro. The con is, you can keep getting factors and factors and factors.
Yes, we have various methods.
It doesn't, you know.
Yeah, so we have 3 methods for choosing the number of factors. You weren't a reviewer for our paper, were you? Yes, we demolished that reviewer. So and we already had two methods. So I mean, this is a problem with any unsupervised data analysis algorithm, right? Whether it's clustering, how do you choose the number of clusters? Whether it's principal components, how many components do you keep, and so on. There's several methods you can use. You can look at the reconstruction error as a function of the number of factors that you keep, and you might stop when you get diminishing returns. You could look at the similarity of the factors that you get, because this is a non-convex optimization problem. So if you start from different initial conditions, you may get slightly different factors. So you could run it multiple times from different initial conditions and align the factors and see how similar the factors are. As you start including more and more factors, they'll stop being similar to each other. The other thing you can do is you can do cross-validation. You can hold out stuff in the tensor, okay, and do the decomposition only on elements that you keep and ask how well you can predict the other elements. All 3 methods are great ways of choosing it. But actually, the other thing is, as usual in unsupervised dimensional error reduction techniques, this number of factors that you keep allows you to trade off between 2 very important variables, the complexity of the model and the interpretability of the model. And we just observed that the answers that we got were pretty robust as we changed the number of factors. Usually what happens is, as you start increasing factors, individual factors split into 2 factors and so on, while all the other factors remain the same. Yeah.
You start off by saying this is monkeys learning over time. Monkeys get faster, so the number of samples goes away. It changes dramatically.
Excellent. So there's various ways to deal with that. You can time warp the data. You could keep roughly the fastest trials and truncate the data, and so on. There's various ways. You can align the data to interesting events and keep fixed amounts of data around the interesting events, like movement onset or something like that. Neuroscientists have dealt with this time immemorial because every individual trial is a different duration. And those are the kind of the techniques that people use, and we use those same techniques.
So you don't do anything like actually, the other thing is that not only is it's not a smooth expansion, because the monkey might get If it's a complex problem, the monkey might do the first part of it fast and then slow down.
Okay,
And
so
all
let's
the time.
jump to the monkey, because we did this on the monkey. So let me tell you what, we did this, so we did this in an artificial recurrent neural network, and it told us how the network learns. We're actually using tensor components analysis to analyze learning dynamics in deep networks, actually. And it seems to yield very interesting results there. We also applied it, do I have that slide? I don't have. Okay, we also applied it to data from Mark Schnitzer's lab, also here at Stanford, on mice navigating a maze, and it yielded interesting effects in the mouse. But let me focus on the monkey here. So this is an interesting experiment. How much time do I have?
It's got a clock right above you.
Right there. Okay, so let me try to do this in 3 minutes. And I can stay afterwards for some questions. But of course, nobody needs to stay. Here's the basic idea. This is a BMI perturbation experiment. So what they did was they had a monkey reaching, and they developed a brain-machine interface that translated neural activity into the intended direction of movement of the monkey's arm. Then they tied the monkey's arm down, and they had a neural cursor. They had the monkey's brain directly control the cursor. So because the BMI was trained on the monkey's natural reaching patterns, the monkey could control the cursor. So if it was asked to reach here, the monkey could think and make the cursor move here and so on. Okay? And these are individual trajectories to a particular target. And you can see the trajectories are not bad. There's some overshoot and return and all that, but it's sort of working. Then they do this vicious perturbation where they rotate the cursor angle 30 degrees compared to what the monkey is expecting. Okay? So now, like if the monkey's thinking that he wants to move the cursor this way, the cursor will actually move this way. Okay? It's a vicious perturbation. Monkey doesn't get it initially. Right after the perturbation, the monkey's still thinking the cursor should move this way, but lo and behold, the cursor moves this way because the experimentalist tricked the monkey. And then over some late time, the monkey makes a correction and then moves back to here. So these are individual trials right after the perturbation. But then, at much later trials, it starts to get better, and it adapts. The monkey's brain adapts or learns to deal with the rotated BMI, right? And so the monkey starts to do more straight reaches, okay? Now, we want to discover what's going on in the brain during this. And of course, individual neurons are super complicated. It's hard to understand by looking at them. So we just threw the neural data tensor, which is, again, order 100 neurons by 600 milliseconds by about 100, you can see about 100 trials, okay? And we found that 3 components, by the various measures of choosing the number of components, were sufficient to explain most of the variance in the data, okay? And so we recovered these 3 components. So this component number 1, 2, and 3, okay? These are the different cell assemblies involved in each component, but let's look at the dynamics, okay? So the interesting one is across trials. So you have some trials before the perturbation, then a bunch of trials after the perturbation, okay? So there's some component that's active before the perturbation, and so this is like the baseline state of the brain, okay? And it's active very, very early in the reach. It's active super early, so it's implementing this kind of movement, okay? Interestingly, the baseline state of the brain, which was good before the perturbation, becomes maladaptive or bad after the perturbation, but it doesn't get extinguished. The brain keeps it around. What instead happens is 2 components are born after the perturbation, and they have different dynamics. One gets active right after the perturbation, and within a trial, it's active very late. And what that's doing is it implements this late-stage correction. But then over time, the late-stage correction dies down, and this early-stage correction or mid-stage correction gets activated by the brain. And it's active in the middle period, right? And it's presumably implementing this and compensating for this thing, right? And so that's how the brain is kind of doing it. It has these 2 components that mediate the learning. Now, remember that this was just extracted completely from the monkey's brain. This data analysis algorithm never had access to the behavior. Nevertheless, do individual components predict anything about the behavior? So as you mentioned, As the monkey gets better and better, it gets faster at the task, right? These reaches are pretty slow because it has to do a long correction. These reaches are faster. So one behavioral correlative learning is the time it takes to acquire the target. So this is just behavior, okay? This is on each trial, how long did it take to acquire the target? It's doing really well and fast here. After the perturbation goes up and it comes down. This looks a lot like this component extracted from the neural data, okay? They look strikingly similar. It turns out that they're actually quite identical on a trial-by-trial basis. So you can try to predict reach time from this neural component directly, and we did that. And now this is data from across all 8 directions, okay? The black is behavior, time to acquire the target. The blue is extracted from neural data alone, okay? And we're not doing a regression because there's just a single factor. We're just scaling one amplitude factor up to match best as possible the time. And you see there's a lot of covariation across trials. So this is what I mean by tensor components analysis does demixing. I didn't show you this, but in many, many data sets, individual factors tend to have a highly interpretable meaning that can often relate to a quantitative aspect of behavior. In this particular case, there's one neural component, right, which is a corrective perturbation, whose amplitude dies off, and it predicts the time it takes to do the reach. Okay? And that's it. So that's the end. Again, Peyran Gao did a lot of the work on the single trial theory, and Alex Williams did a lot of the work on tensor components analysis. We actually have lots of collaborations, lots of experimental labs, and we're also working theory of deep learning and stuff like that. But again, thanks. All right. Any questions?
So have you tried anything other than monkeys?
Yeah, we actually work with mice, with flies. We even have some work on humans. What else? Salamanders. Yeah, we're working on a bunch of things in collaboration with experimentalists. Yeah, different animals are better for different questions. Was there a particular?
No, I'm just curious.
Yeah.
How do you? There are presumably ethical issues with working on people. How do you? What's the?
Oh, with people, it's more like psychology data that we work with. So we have some work on like theories of how infants develop concepts. And there's all these psychological studies of when infants can learn categories and things like that. And neural networks learn categories with a certain dynamics. It turns out neural networks and infants learn categories with the same dynamics. And we have a mathematical theory for why. So with humans, it's mostly psychology data or fMRI data, but I've kind of stayed away from fMRI data, just not because it's not interesting, but I'm just swamped with electrophysiology data from my...
Can you get actual human data under the right circumstances?
Yeah, usually, so there's various modes for measuring data from the human brain. There's fMRI, there's EEG. Those are non-invasive. Then there's ECOG, which you can often do in, say, epilepsy patients by putting surface electrodes on the surface of the brain, so you don't get low-pass filtering through the skull, which you would get on surface EEG. That's done by, for example, Joseph Parvisi here. We've looked at some of that data, but, and then you can even put in depth electrodes into the human brain, again, only in patients that require that kind of monitoring of the brain, say to excise some part of the brain for epilepsy or something like that.
They're putting probes deep in Parkinson's brain.
Yeah, so deep brain stimulation for Parkinson's, that's another method for treating Parkinson's. That's definitely something that's done. But it's very hard to get multi-electrode data from the human brain, though that is being done by Krishna Shenoy and Jamie Henderson here.
So I don't have a picture of how your probes go in. How do you get 100 probes into something?
Oh, there's various ways to do this. There's actually some new technology called neuropixel probes, which is this linear array. And you slowly squish it into the brain and wait for the brain to stabilize. And then you have electrodes all along the array, right? There's also surface electrodes, which only allow you to record from the surface of the brain. And it's a bed of electrodes, a 2D array of electrodes, and you stick it in a little bit into the brain. And you
tiny micropipettes with electrodes.
Yeah, exactly, yeah. There's also calcium imaging where you can record fluorescence coming from, so calcium rushes in, that gates an ion channel, that gates a particular molecule to fluoresce, and you measure the fluorescence. That's something that happens as well.
With these multi-electrodes that you do surface of the brain type of stuff, never personally used them. I've used different, but how many hours or how long a period of time can you keep?
Oh, that's an interesting question. Yeah,
From
it varies from technology to technology. For some, you can keep them in for months, but there's no guarantee that you'll get the same neurons on day 1 versus day 2 versus day 3. There's some churn, and people believe to different degrees claims that you got the same neuron over multiple days. If you want to do that kind of stuff, you do calcium imaging, and that you can stably get neurons over many days. That's the same neuron.